moving right along. So yesterday we set up these problems, and today we'll set up some more problems in here. And then you're just going to have an activity really that's getting ready for what we learn next, just to take a break from doing problems. I'd much rather color than I would like to do genetics problems. I know you probably don't believe that. So the following pedigree shows inheritance of 2 human genetic disorders. Symbols with vertical stripes, so these indicate individuals who have, I can't read that, I gotta put my glasses on. Okay, so it's alkaptonuria. I still can't read that. Wow, that's disturbing. I guess I will have to get new glasses, which is inherited. And then horizontal stripes, so these have tyrosinemia. But it's an inherited, does this one say enzyme deficiency? Okay, let's try a different set of glasses. Go back to my ones that are the newer ones. Okay. Oh, wow. That's much better. Okay. So you've got a metabolic disorder and an enzyme deficiency. And you've got families who don't show the disease, who have children who have the disease, and then these children marry spouses or mate with individuals who have the other disease. And so at this point, based on the information here, is, let's just call this A dominant trait or a recessive trait, and give evidence. This is where you're going to do the same thing you did with pedigrees A, B, C, and D, where you go through and you look. So this is the one that's vertical stripes. And there should be, in your mind, a real quick, easy answer with that. And then let's use T's. So tyrosinemia, is that a dominant trait or a recessive trait? Again, give evidence. And what's the chance that the couple at the bottom of the pedigree will have a child, indicated by the question mark, who has both? And so you're going to come through here and you're going to have to recognize what these individuals are. And you're going to have to write that with both A's and T's. So, for example, this person here has, which one? Horizontal tyrosinemia. Okay, so I know if I said it was recessive, they would look like that. But they do not have the other disease, right? The alkaptonuria. So all I know is this. And then you'll just work your way through, okay? And answer the question. So this is a you do. I'm going to circle the ones. Ooh, guinea pigs. You buy a cream-colored guinea pig and you like its color so much, you're going to breed it and sell the offspring and have guinea piglets. So you buy a second cream-colored animal, but woo, to your surprise, this sounds very much like the purple ketchup and the pink flowers and the, what did we just do here with, I don't know. Oh, the chickens. You end up with 15 cream, but 8 yellow and 6 white. So that looks very 1 to 2 to 1 to me. So I'm going to leave you with how the color is inherited. What they're looking for there is complete dominance, which means you would have 2 phenotypes. Incomplete dominance, where that you have 3 phenotypes with the third a blend, or codominance, where you have 3 phenotypes and the third is like stripes or spots or blotches or patches. So you do this one. In mice color, coat color is determined by gene B, which has black and brown alleles. Black is completely dominant over brown. However, there's a second gene C which affects mice. We've done this problem. Mice must have at least a dominant allele. So this is, you want to go back and look at 10, 11, 12, because this is epistasis. Because whenever you see that it has a gene that has to have this regulatory gene as a dominant allele, then if they don't, then they're white. Okay, so off you go with this one. We're almost done with this packet. These are much more like AP level questions. Okay, where's your number? What happened? We went from 24 to 30 something. Oh, that's good. Certain kind of flower is red because red pigment that requires 2 different genes. Gene A encodes, this is right here, the enzyme that catalyzes the conversion of something colorless into this intermediate colorless molecule. So it goes from colorless number 1 to colorless number 2. Then there's another gene, gene B, that codes for an enzyme that converts this second colorless number 2 molecule into red pigment. Both have to be working. So you have to have dominant alleles for both of these genes so that they create the working version of the enzymes for both of them. Okay? I don't know what number you are. I'm going to give you the number 812 because obviously we don't care about numbering because we're all over the place. That's a you do. While working at your summer job as a campus groundskeeper, you gotta love humid summer afternoons. You notice that all the ordinary short yellow marigolds are out there, but there's this proud, tall plant with bright red flowers. Thinking you could make some money selling this unusual plant, you do a cross of the tall red with a short yellow. Okay, short yellow, we're thinking was the common one, right? And the tall red's the one we want to reproduce. Oh, you're so disappointed. In fact, you don't get any red at all. Only some of your offspring are tall. So looking just at height, think about what would happen. Looking at color, think about what happens. And then they want you to grow and have seeds that are only tall and red. Now this one's more difficult than the ones you've done, but you can do it. So that's a you do. I'm through with the you do's. Okay, I think we have one more page left in there. So next, you should have some drawings that look much nicer than this. Much nicer, but these are the ones I have, and this is what we're going with. So we're going to color in what we can color so that we're just not learning, but we're coloring. Let me find my paper. I'll be right back. For half size. And when we're done, we're going to have all of the process of making a copy of one strand, double-stranded DNA. So you'll end up with exact copies and you'll have 2 double-stranded DNA because DNA is double-stranded. So I think what I'd like for us to do is to try to make some of this as simple as possible. The easiest things to do are to, you need, well, how's that going to work? Where's not? There it is. You'll need an orange, and don't go get any because I'm going to tell you what you need. And a green. That's the only green I got? Okay, fine. And you'll need a navy. Is that navy? That doesn't look navy. Nope, it is. And a red. And probably a light blue. And that should do it. We'll draw some things in that we'll leave blank. But right now we're not learning because we know we can't do both. We're just coloring. So let's work on your papers. Now you're going to have these on 2 separate pages because you need a whole bunch of space at the bottom to write the kind of stuff that we're going to write about each one of the pictures. And then the next set of pictures end up looking like this. They're gorgeous. But for now, we can just load a couple of these and get started. So, okay, what's up with the paper? So on one, up here where that you have, let me shut this blind. Better? Yes. Okay. So all the time, this top strand right here is going to be orange. So you're going to do that better than me on all of the pictures, every single one. And then if you think about where this would go, it would dip down and go like that. And then for this bottom strand, go ahead and make all 4 of them green. Okay, so you're going to do that on all, make sure I'm right. Yep, all 4. The other thing you're going to do on all 4 is you're going to take red, and that looks like teeth, but they're not. These are the nucleotides. All of the long ones, you're going to color red. The red are the bigger ones, and if you remember, the stupid thing, the way we learned it was, it's not fair that you get the big name and the long length. So if you get the big size, you have to get the small name, which is purine. And if you get the smaller size, which are these shorter ones, then you're a pyrimidine, and they're going to be navy. So that's what you're going to do to get ready for Tuesday. So off you go, and I think that we'll just do what we've done here on all 4. The only other thing is this is a molecule called helicase, so it's an enzyme because it ends in ase, and its job is to pry open and stay where this is called a replication fork. So it's like a wedge, and it pries open these 2 strands so that you can make copies, 1 copy of the top one, a copy of the bottom one, and then you end up going through that whole synthesis part of the cell cycle where you duplicate the DNA. So off you go. That's all you've got. So you have hopefully got time, this shouldn't take that long, to get these prepped and then to finish the problems assigned to you. Again, let me stress, I'm going to take those problems and they're going to become part of our notebook check, but not just because you got something written there. Okay, so if you wrote 1/8 for the answer for the, I don't know, right, the proportion for having both diseases for that one question, that's not going to get you anywhere. You're going to have to explain it and be able to understand it, because, you know, this isn't just about having a notebook we copied. This is about having practiced the work. So my fear is that this next notebook check could become a grade that's not what we're used to. Like it could hurt you instead of help you. So be sure you do your own work. Be sure that you can explain it when you have a notebook check. And then off we go, because as soon as we're through with this piece, which I'm thinking we could probably do Tuesday, Wednesday, Thursday, Friday, you're looking at another practice test that will be the actual genetics test. So we got a lot we're doing. Got to get ready for May. You're amazing in what you're doing. So just learn. Don't be about finishing. I'd much rather you not have finished something and that you need more time because you've honestly tried than to have copied it from somebody and you have no clue. So if I find that, I might have to selectively give quizzes with problems that look like what we did. I won't give them to everybody, but people who just have answers, they'll have to take a quiz in order to demonstrate they actually did the work. But we'll figure it out. I'm not trying to be mean. I love you. So this is it for Friday. I hope you have a wonderful weekend. I hope I have a wonderful weekend. My goal, the big deal is stupid headache. It's awful. And the fever, which I don't ever have. So I'm super grumpy. Better be glad maybe I'm here and I'm not there. So, but anyway, I love you. And we will pick this up on Tuesday.